thank you all for those of you who braved the heat and the humidity and the traffic to be here. And for those of you who are at home in your air conditioning in your pajamas, you don't get as much credit. But glad you could join us. I've been asked to sort of give a primer on clinical trials or clinical research. And I have a lot of text slides, which is just kind of boring, so please interrupt me, and we'll try to keep it lively. So, what is clinical research? So it's research with human volunteers, and we call them participants or subjects, which is always kind of funny. We try not to call them patients because we're not actually delivering care during the visits. And these are very carefully conducted investigations to ultimately uncover better ways to treat, to prevent, diagnose, understand human disease. Human subjects have certain rights, and these are laid down in agreements actually between countries, not even just in our own country, so Helsinki agreements. And one of the ways in which human subjects are protected is by the study design. So there are different rules, for instance, of what we can do in our studies depending on the age group, for example. So for children actually have to have the potential to benefit if we're going to give them more than minimal risk. That's not true for you adults. You can be exposed to risk. You can choose to be exposed to risk when there may not be a benefit. We have things called institutional review boards, which you've probably heard of, called IRBs. And these are independent groups of people, committees that consist of physicians, statisticians, and always a member of the community to ensure that clinical trials are ethical and the rights of the participants are being protected. So these people have nothing to do with the clinical trial or the investigators. Another key component of human subject protection is an informed consent. So this is actually a fairly lengthy process in which the participant learns key facts about the clinical trial before deciding to participate. It's the very first thing that has to happen in clinical research involving a participant. And this informed consent includes the purpose, the duration of the trial, required procedures, risks, potential benefits, and who to contact if something goes wrong. And then many studies, not all, but many studies have a data and safety monitoring board. And this is, again, completely independent. It's independent from the investigators. It's independent from the institutional review board. And this group of experts usually have some understanding of this particular disease process or the outcome measures. And they advise the sponsor, the people who are funding the trial, and the study investigators. They're responsible for periodically reviewing all the study data, both the quality of the data coming in and what the data says about safety. And they make recommendations at each meeting. Do we continue the trial? Do we recommend changes to it, or do we say you have to stop? And recently we performed a clinical trial in a different muscular dystrophy here at Kennedy Krieger, and our Data and Safety Monitoring Board thought from reviewing the data that the drug may actually be inducing some harm, and they recommended stopping the trial, which we then did. So that does happen. Sometimes the data looks so good in favor of a drug that the Data and Safety Monitoring Board will also recommend stopping at publishing the data and having that be a recommendation for care. So there are lots of different types of clinical research. So you've heard about one type of study this morning, and so not all clinical research is we're going to give a drug or treatment and try it. So there are observational studies where where the investigator assesses health outcomes in a group of participants. There are natural history studies. Those are incredibly important for muscular dystrophy still. How does the disease or health progress? The natural history changes over time, so the natural history of FSH in the 1980s may not be exactly the natural history in 2014. And the natural history of FSH in the United States may not be the same, probably isn't the same in some other countries. There are prevention trials, screening trials, diagnostic trials, treatment trials, or interventional studies is what we tend to think about when we think about clinical research, but there's so much more. So treatment trials would test a new treatment, a new combination of drugs, or a new approach to a therapy, see whether it's safe and efficacious in a diseased population. And there are also quality of life trials. So I showed this figure at the FSH Society meeting in Boston to just give you an idea of what are the different stages that a drug would go through in development, beginning with animal trials, and then phase 1, phase 2, phase 3, FDA approval, and also showing the number of compounds that are assessed at each stage and the amount of time that it may take for a drug to go through from animal trials all the way up to FDA approval. And many people are surprised at the length of time that it does take. So let me talk to you about each of those phases. You're still with me?
Yeah.
Not that it's complicated, it's just a little boring. So phase 1 is where we assess drug safety and tolerability. So these always begin with healthy volunteers. Well, I shouldn't say always. Sometimes a drug could potentially be harmful for a healthy volunteer, but be the right thing for a target population. If that's not the case, then it begins with the healthy volunteers. And then you may also do a phase 1 in the target population. There's a limited number of people. And in addition to looking at how safe is the drug and how well is it tolerated, meaning, does it give you diarrhea or a headache that may not mean that it isn't safe, but it just is really not something that people would want to take. It's not tolerated. So in addition to that, you look at pharmacokinetics. So how well is the drug absorbed into the bloodstream? How is it metabolized? Is it chewed up by the liver or the kidneys? How is it excreted, for instance, with urine or feces, or exhaled through the skin? And these phase 1 studies are usually dose escalations. So start off with a very low dose to assess safety and toxicity, and then increase. And about 70% of all new drugs pass this phase. So phase 2 drugs assess the drug efficacy, and they're further evaluating safety. These are randomized, and I'm going to talk a little bit about these. They're controlled studies. We look usually at what we call surrogate outcome measures, and I'm also going to explain that. They're relatively short-term, still relatively small numbers, meaning less than 100 patients usually. And about 22%, which enter phase 2, go forward. Uh-oh. Okay, I have my slides a little out of order. Phase 3 are large-scale randomized controlled trials to confirm efficacy and safety in a large population. So this would then use hundreds of patients. So, for instance, there's a trial right now. Well, there have been a couple trials in Duchenne that have used around 300 patients. So those are more phase 3 trials. And they tend to be longer term, month duration, and try to use outcome measures similar to the real world, to symptoms that someone is having at home, such as function or quality of life. This will define what's in the package insert that you get when you buy any drug, and it will also define how the drug is marketed. Is this marketed for a specific disease? So, in the clinical trials, if you try something in Duchenne, it's not marketed, it's not FDA approved for all muscular dystrophy. It's marketed for and FDA approved for Duchenne or for FSH. And 55% of drugs succeed at phase 3. But if you multiply that 70% by 22% by 55%, you'll see that only 8% of drugs actually make it all the way through them. So 8% of drugs that enter clinical trials make it through to FDA approvals. So most clinical trials fail, or most drugs fail in clinical trials. So what is randomization? You may be more sophisticated than the average person who comes into a clinical trial to participate, but many people don't really understand that whether they receive a drug or placebo or a particular type dose is completely random. That if you are my favorite patient, I can't put you on the drug. It is done by a computer-generated model. So I brought an example here. Let's say that we're going to do a trial of 40 people, and we're going to randomly assign them to 4 different treatment, what we call arms or groups. So condition 1 is that we've got wonder drug that's going to be at 5%. Condition 2 gets wonder drug at 10%. Condition 3 gets wonder drug at 15%, and condition 4 is placebo. So we assign each participant a unique participant number. It's usually the number which they enroll in the trial. And then we use a randomizer algorithm, something on a computer, to generate a set of 40 non-unique, unsorted numbers with a range from 1 to 4. So the first person that enrolls, I just put this in the computer this morning. So yeah, it was this morning. This was kind of last minute talk. So I just put this in and to randomly generate these numbers. So the first person I enroll, 3, they get wonder drug 15. So I guess that was my favorite patient. But I really liked the next one who I enrolled, my second subject. Unfortunately, they got placebo. So that's what randomization is, completely generated by a computer and assigned. And usually this code is kept by the pharmacist. So I don't actually even see it. The pharmacist is assigning the participant to the particular drug based on this code. So what is a control? And we've already been talking a little bit about that. Do you like that? Do know right about that. A comparison group that receives a placebo or another treatment, or no treatment at all, which is not the best type of control. But very frequently in our drug trials we use placebo for muscular dystrophy. We don't have a lot of other treatments to compare to. So this doesn't have to be a 1-to-1 ratio, as in the example with the randomization. The placebo group was only 1 out of 4, and it frequently isn't the case. Okay, so there is also a phase 4 phase to clinical trials, and this is after the drug has been approved by the FDA, and so it's on the market, but we're continuing to gather data on it, and the drug may be compared to other drugs on the market. FSH should be so lucky. We should all be so lucky that we could have a phase 4. We're really not going to be at that stage. But for instance, if you're studying hypercholesterolemia, increased cholesterol in the bloodstream, then we have multiple different cholesterol-lowering agents out there, and so you want to do a phase 4 study to show that your drug is actually the best. And you'd be monitoring also long-term efficacy and safety over years. And sometimes with the phase 4, drugs are pulled from the market. So who do you meet in a clinical study or in clinical research? The dream team. So usually there's a principal investigator. Well, there is always a principal investigator. Usually it's a doctor. And they may have co or sub PIs. So in studies here, I'm frequently a PI, and we have Carla Grossman right there. And we have Doris Leon, who are both co and sub-PIs with me. And a clinical trial coordinator. This could be a nurse, a doctor, or other professionals. They're people who really study how to do clinical trials well and have experience in this. They make sure that the trial runs smoothly operationally, and we have Jenny Bubat. And then there are clinical evaluators. These are frequently in neuromuscular diseases. These are frequently physical therapists. They may be something else in other disorders. I don't know anything about muscular dystrophy. So we have Carly and Amy here at Kennedy Krieger. They measure muscle function, and we were learning earlier about specific ways that you could measure muscle function. And they can be ways that measure lower extremity function, such as walking, but they can also be ways that measure upper extremity function, such as the 9-hole peg test that you mentioned, I believe. Then usually there is a nurse as a member of the study team who collects biomaterials, urine, blood, etc., and very frequently administers treatment. So the study design is really key. That's where a lot of the intellectual process of the clinical research comes in. Really, the how to design a study to really ask the question, a good question, and ask it correctly. And a lot of drugs fail, not just because the drug isn't good, but because the study design wasn't good. So clinical research is conducted according to a very specific protocol that's going to say who's eligible to participate, what the procedures are going to be, so you ask what would actually happen at each visit, what the length of the study is, the number of visits, what information is going to be gathered at each visit. So it starts off, your study starts off with a screening, and as I mentioned, it always starts off with an informed consent, and then a determination of eligibility. And there are very strict inclusion and exclusion criteria. There are a list of you must have these things and you can't have these things. And we learned about that for the infantile onset FSH study. And the reason that that's so important is so that we're not comparing apples and oranges, and that we have a group of people who we anticipate that will actually be able to measure what we're setting out to measure. So these inclusion and exclusion criteria would include age, gender, type and stage of disease, previous treatment history, other medical conditions, other medicines that someone might be on. One of the things that's interesting for neuromuscular diseases is that we also really very frequently have 2 screening visits in which we look at what we call our primary outcome measure. We look at some particular function, for example, and we want to see that it's reproducible from screening visit number 1 to screening visit number 2. And that's, for instance, let's take young kids with a muscular dystrophy. You know, maybe one day they're really motivated and the next day they're really not motivated, and those 2 values are wildly different. Well, we're not going to be able to say whether our drug is working if we've got that much variability. So having a lack of variability is important. So there'll be multiple visits, usually not always. In the infantile study, there's a single visit. We have a bone health study with FSH. It's a single visit. But if you're doing participating in a treatment or interventional study, there will be multiple visits. And you'll get a schedule that might look something like this. This is from an old study that we did, in which you'll see that on various different days there are various different activities. You start off with an informed consent, and we look at inclusion and exclusion criteria, and we get some baseline measurements. And in other visits, you'll be also being assessed for safety, and you'll be getting the drug, etc. And you'll get a schedule such as that in your informed consent. So outcome measures, I've mentioned a couple times. What are these? So ideally, they would be measures that are meaningful to patients' everyday life. So, for instance, if a drug changed longevity, let's take ALS, amyotrophic lateral sclerosis, that would be, an extremely meaningful outcome measure. FSH, we don't have major changes in longevity, so something that changed your motor function, your ability to do things. Other measures, however, are ones that correlate with those very meaningful measures, and we call those biomarkers. So this is an example of a kid participating in a clinical trial where we use the 6-minute walk test. And that's being considered for FSH, muscular dystrophy too. Obviously that would only be a valid outcome measure if we were looking at a group of people with FSH who were ambulatory. And so in this test, we ask people to walk for 6 minutes, and we see how far they can go. And there's a lot of interest and emphasis now at FSH in being able to identify good outcome measures, because that's really not known. And if we have the wrong outcome measure, then we could potentially ignore the right drug. And so there are efforts here at the University of Rochester, Ohio State, to measure many different things and see what are good outcome measures for people with FSH. And so we have been looking at 6-minute walk test here. We've been looking at time to up and go. How much time does it take you to rise from a chair and walk? And can I say your name? And so Kelly has participated in that, and I think other people, Jim has participated in that also, and Lillian, lots of you. I mean, I really can't say enough that, you know, how wonderful it is. The FSH population always turns up, and you know, you say we're going to do a study, and people sign up to help, and it's really great. So we are collecting this information, as are other groups. There's a really nice quality of life questionnaire that has been designed by Chad Heatwall that looks at has a questionnaire to look at things that are meaningful for those with FSH, and that may be a really good outcome measure also. So you do participate in a trial. After the trial, what happens? So the information is collected and is studied. Well, the information was collected and is studied. And again, the beginning and the end, in my opinion, was where the real intellectual part of a trial is for the investigators. And a decision is made whether to go forward with the next phase. If this is a phase 1 or 2 or 3. The results are often published in a peer-reviewed journal. They're not always. If it's a negative trial, they used to never be published. And now there's a lot of emphasis to publish even the negative results because people learn from them. It's just not obviously as interesting when it's negative. The results specific to the individual participant are frequently not shared with the participant, and that can be really frustrating, and that can also be something that's not known by participants when they start. They think, oh, all this data is going to be generated on me, and I'll get to see it. Not necessarily. The people who conduct the trial may not even really know what happened to each individual. So for instance, if I send somebody to get an MRI and that data is collected and sent to a central source to be read and compared, I may not actually know the results there. So why would you participate? I don't actually think I need to do this too much with this group. You know, I think you're all really very motivated. But you know, playing an active role in research and improving the treatment of the disease, I have come to see how important that is to people. That to hear about research is not as rewarding and not as affirming as actually participating in it. And I see that a lot. Many people do participate to gain access to new treatments before they're widely available. That is a real motivation, and I think particularly, you know, if you have a severe disease, let's say you have cancer, and you know all of your standard therapies are unlikely to work, then you may want to participate in a clinical trial, even though you know that it's not a high likelihood it may save your life. What I usually tell people who are participating in trials in neuromuscular is that you should do it for the first reason, and not so much for the second one, because most trials fail. And I think if you go into it thinking, I'm going to participate with the hopes that, you know, my particular muscular dystrophy is going to get better, then most likely, you just, you know, the odds are, most likely you're going to be disappointed. And the third is also a real feature of trials that's been documented, that people who participate receive a lot more looks at them. They receive a lot of medical attention. Now, maybe you don't want all that medical attention, but other findings can pop up, and it can be helpful. So, Lillian, can I mention your recent? So, Lillian has participated. I'm just blowing all sorts of HIPAA guidelines. Lillian and several others of you in the audience have participated in a bone health study that we're doing in FSH. And so one of the things that we're finding is, people who participate in this bone health study, they've got low vitamin D, and we call them up and we tell them that. And Lillian had participated in it, had a DEXA scan done, and then promptly went out and got a fracture. But then she's had all that work done. Then boom, she's plugged into an endocrinologist to get appropriate treatment for below bone density. So I do believe that there are a lot of advantages to just all that medical attention that you receive. Questions that you want to ask as you're considering any type of clinical research, particularly a clinical trial. Who's sponsoring the trial? It's very different if it's investigator-initiated. If I say, I'm interested in running a clinical trial with insuldenafil versus Pfizer says, we want to run a clinical trial in the drug that we market. What's the participant burden? And there are all sorts of burdens on your time, on your money, on how you're feeling. What are the risks, not just of the drug, but of the various different studies that are going to happen? So if you're going to get an MRI with gadolinium, what's the risk of that? And then in terms of the risk, what's the degree of harm that could be expected? So for instance, a muscle biopsy, very frequent in clinical trials with muscular dystrophy. Well, yeah, there's risks in a muscle biopsy, but the degree of harm is incredibly small. Maybe get an infection, maybe get a bruise, but really very low degree of harm, not life-threatening risk. And then what's the chance of that harm too? So let's take the muscle biopsy example again. It's less than 1% of people have any side effects from a muscle biopsy. I think you should ask what's going to be billed to me, my insurance company, versus what's going to be billed to the study. I mean, it should actually be in your informed consent, but you should understand that. What is the ratio of placebo to treatment? Is this a 1-to-1? Is it a 1-to-4? Maybe there's no placebo group. In some phase 1 studies, there was no placebo group. And is there a commitment to an extension study? And people who participate in the Maya 29 probably have some opinions about that. So people who participated in the anti-myostatin treatment for FSH everyone was hoping, and many were expecting that there would be an extension study, that there would be a subsequent study in which they were put on drug while the drug continues to be developed, while we're waiting for FDA approval. And that didn't happen. And the reason it didn't happen is that the drug didn't go forward. There were no subsequent studies. So if the company just says, we're no longer interested in this drug, then there's no extension study. And that can be extremely disappointing. So why do clinical research in FSH now? Again, I know I'm preaching to the choir a little bit. So, the medical community learns about FSH in each of these studies that we're doing, and that moves the ball down the field. The research community also learns just how to do these studies. It's not the same. Doing a study in Duchenne doesn't necessarily make us good in doing a study in FSH. They're different. The biomarkers are different, imaging is going to be different, endpoints are going to be different, and so we need to actually do studies in FSH. And the patient community becomes really very engaged and motivated, and this is our favorite Don Burke here, and I love this a quote of his, which I've used before. He says, "To my surprise and appreciation, I found myself immersed in a medical system that overflowed with passion for its work and for FSHD. I was surrounded by doctors, surgeons, nurses, and aides who are attentive and engaged. I found myself looking forward to each visit, even the muscle biopsy, and exchange of information and knowledge. So,
quite the surreal experience.
So I think that's all I have, but I'd be happy to ask or answer questions.
Could you comment about being involved in multiple trials at once and the importance to the people conducting trials, that we make sure that we tell you whether or not we're on a treatment trial or another kind of trial, so that we're properly either included or excluded?
Yes, sure. So the question was to comment on participating in multiple studies at once and how we communicate that and the importance of communicating it. First, I should just say we should be so lucky. But for treatment studies, if you're going to be involved in a treatment study, you usually are committed to just that one study, because that you can't be in two treatment studies because of the potential side effects for one drug may be used as the side effect of the other drug, they may interact, etc. But also, it's very hard to be in a treatment trial and an observational study, because the treatment may influence the natural history of the disease. So it is extremely important. Let's say you were involved in Doris's longitudinal MRI study, and Children's National starts a nutritional supplement study. You know, it's really very important to notify Doris that, you know, well, do you still want to look at my MRI of my thighs or whole body, because now I'm doing something that may alter the course of this disease. You can very frequently be in more than one type of observational study, however. Jim?
Could you expand on the individual information? I understand why I can't get my own data, but I would really like some feedback on, hey, we published this paper and you were in it. I mean, I was very interested in in being a volunteer, but I'd also like to know that, hey, I'd like to read the report. Yeah.
So the question was, if I could rephrase that a little bit, the question was, why don't we get the information, and why don't we get more of it, and why don't we at least get the published report? And you should at least get the published report. And I think that investigators may not be as good at doing that as they should be, myself included. And so it's very reasonable to remind them that that's something that you're interested in. When I think back to the Mayo 29 study, there was a huge lag. I mean, 2-3 years from when the study ended to when it was published. So, you know, I'm already moving on, and Mayo 29 wasn't getting developed. So in some ways, you know, we didn't do a good job of relaying those results. What we did at our site was have a reunion of the participants, a little wine and cheese. So we talked about the data in general, and this was before it was published, however. And I don't know, did we give out the patient information?
If they wanted it.
Yeah, right. So, and whether the participant gets to know whether they were drug or placebo also depends somewhat on the sponsor. So there, no, it's all initially double-blinded, meaning that neither the participants nor the investigators know who's on drug. But at some point, the investigators usually do become unblinded. They don't always. June and I and my husband were looking for jokes to liven up my text slides this morning, and yeah, we found a couple, but I just have to share one with you that I didn't put in it. So what is the definition of a double-blinded study? It's two orthopedic surgeons trying to interpret an EKG. That's my survey bits.
And you shared that with your medical school friends?
Hopefully Leanne Curl, my favorite orthopedic surgeon, is watching right now.
I sort of have a series of kind of related questions. Just first, the quote that she pulled there is from an article that's in one of the past FSH watches. There, I think myself and a couple others can convey what our experience was with this trial. What are some of the reasons that cause so few drugs to get through? Because I mean, they cost a lot of money, and clearly people aren't going to go into a trial without there being pretty good evidence that there's a reason to try that trial. Well, why is it that we only end up with 8% out, even in the modern day of medicine where we have really good, mathematical models and pretty good animal models in many cases? What causes that winning?
Well, that's a really good question, and so the question was, why do only 8% of drugs that enter clinical trials get FDA approved? And certainly investors would like to know that. I think that there are at least three main reasons. One is that our preclinical models are not predictive, and maybe they are. I mean, I do studies on rodents, and so I don't want to throw them all out. But maybe they have a particular role that we have now expanded widely and inappropriately. But it's very clear that most things that work in rodents don't work in humans, and so that leap is huge. The second is that there are poor study designs, and the MAIA-29 trial has been heavily criticized for its trial design. So the MAIA-29 trial was a phase 1/2 in which it was both looking at safety and hoping for efficacy, but it was also hoping to be what's called a pivotal trial, which was that it would lead to FDA approval. So the more subjects that it had with diverse disorders, the more likely they could say this is a drug for FSH and for limb-girdle and for Becker, all on the marketing. So it was trying to do too much and ended up doing nothing. Right. So if it had been a trial in FSH, I think it would have been awesome. I really do. But there were too few people in the FSH category to say anything definitive. The third is that despite really strong preclinical data and really good study designs, drugs do have unexpected consequences, and so I'll take another example of ACE-31, a myostatin inhibitor that, oh boy, that looked great. I mean, a single injection to postmenopausal women, you could measure increased muscle mass, and it was an incredibly exciting drug. And then injected into children caused nosebleeds and telangiectasias, and it's dead.
You want to maybe add to that pharmaceutical companies, even if they had a successful phase 3, still have a huge investment to make to bring that drug to market. And that's also a potential hurdle for many pharmaceuticals.
Good point.
So, why do you think that the placebo, this is talking about the placebo side of the trial, why do you think now it's so much stronger now? I read an article that the placebo in today's society is so much stronger, has such more positive results.
You mean the placebo effect? Is that what you mean?
Placebo effect, yes.
Why is it stronger now than it used to be?
Yeah, they're seeing in these different trials that placebo has much more of a positive result than it ever did in the past years.
So to repeat Lillian's question that I don't know the answer to, is why is there a stronger placebo effect now in clinical trials than perhaps there was in the past? And I don't know. I'm not even aware of that. But the only thing that I can say is that in my experience, people who participate in clinical trials are very motivated to get better. And kids who you wouldn't even think really understood their disease that much, they're really motivated. Adults who do understand, they want to get better, and they want to believe that they're on the drug and that they are getting better. So I certainly understand the placebo effect. Why it's stronger now than previously, I'm not aware.
So up in Boston, you were sort of lamenting the lack of clinical trials currently underway in comparison to even other similarly rare diseases. Do you think that we're entering a phase where that's likely to change, or are we still sort of in are we still 5 years out from having a lot of clinical trials?
We might be 5 years out. So the question is, is the pace going to pick up? Are we going to start having more clinical trials in FSH? Yes, I think so. I don't know if we're going to be having a lot in 5 years. I certainly hope so, and I do believe the pace is going to pick up, and I think there are a number of reasons for that. The clinical research community is certainly more organized for FSH than it ever has been. The FSH Society is sponsoring a meeting this spring, May, in which many investigators will come and discuss clinical outcome measures. We had a first one in Leiden, and this will be a second one follow-up from that. We're collecting data. So I think that we're going to be more organized and more knowledgeable about how to do a good clinical trial in FSH. And then understanding the pathophysiology of FSH opens up lots of possibilities. So we're not going to be having to, it's hard to get excited about doing a clinical trial on creatine. You can take creatine if you want to, and if it gives you abdominal bloating, then stop. It's just, you just can't get that excited about it, or convince somebody to, give you a lot of money to run that trial. But if you've got a DUX4 inhibitor, then, that really is something you could raise money on to have an expensive clinical trial. So understanding the pathophysiology will help. And then thirdly, there are lots of good animal models and cell models to screen for new drugs. I talked in Boston about a xenograft model that we have at Kennedy, and that we're sharing with other people, in which we take bits of muscle from FSH and grow them in mice, immunodeficient mice, so they don't reject the graft. And this xenograft will really allow us to test compounds, one of which we're doing with Genzyme.
I want to just mention to the group how they can look up what trials are going on trials.gov.
Good. So Carla brings up that all reputable trials, all trials in the United States and all reputable trials in Europe are listed on clinicaltrials.gov. So you can go to that website, you can put in FSH, or facioscapulohumeral muscular dystrophy, because there's also a hormone FSH, follicle stimulating hormone, that you're not interested in. And I guess unless you're trying to have a baby. And that will bring up almost nothing right now. But look at it every 6 months and see what studies and trials are available. Yes.
You mentioned creatine. I had heard that is possible help.
Yeah.
Can you give me a list of things that possibly can help, even though there's been no clinical research that are over the counter?
Yeah.
Or nutritional supplements?
So the only two that I routinely talk about in my clinic, and I think you'd get many different opinions, and if you'd like to also offer your opinions. The only two that I mentioned in my clinic are coenzyme Q10 and creatine. They're both really benign. You're not going, if you have normal kidney function, you're not going to have any long-term or even short-term consequences. So creatine very clearly increases muscle mass and strength in normal muscle. And my feeling is that for many people with FSH, most of their myofibers are normal. You know, you look at it under the microscope, it's not full of a lot of degeneration, fibrosis, scar tissue. Most of their myofibers are normal. So you give it creatine and they will enlarge. And that enlargement of the myofibers may lead to some small improvement in function. That's my rationale. There's no clinical data at all. Coenzyme Q10 is also extremely safe. There's accumulating data that in other muscular dystrophies that it's helpful. Again, never been trialed in FSH, but I think that's also a very reasonable thing to take.
What type of doses?
So for creatine, for an adult male, I would take 10 grams a day, and coenzyme Q10, I would take 300, 400 milligrams a day. Would you like to comment?
People have also tried albuterol for FSHD, but they've been shown to have some improvement in grip and flexion, but I don't know if it's long-standing.
Can you repeat that?
Yeah, so the other comment was albuterol. Now, albuterol is a prescription medication, but it did in clinical trial FSH show us some benefit in isolated muscles.
Possibly there's some muscles that are not affected, so those two supplements could potentially help the muscles that are not affected.
That's correct. Even a muscle that is affected, those supplements may be helpful to the healthy fibers in that muscle. Not all your fibers are degenerating. Let's say you have a very weak bicep, you still got some very normal looking myofibers in that bicep. So, but there's some hand waving there.
I have a very general question to researchers and trials and such. Is there anything, what do you see in FSH as being particularly unique? I think there's some things that are very special about FSH that make it a good research topic.
So the question is, what do I see that's unique in FSH that makes it a good research topic?
Are there things that are unique?
Yes.
That make it a good research topic, and we should get money?
Absolutely right.
That's much to be seen.
FSH is clearly a unique disease among the muscular dystrophies. It is clearly an epigenetic disorder, as we learned about this morning. It's not a single monogenic trait that's inherited, and it does not appear to follow a progressive linear decline as a lot of muscular dystrophies do. I think that is both the excitement and the challenge in studying this disease. So, for instance, if we do a clinical trial in FSH and we want to say that our DUX4 inhibitor is helpful, well, what if DUX4 is only turned on 2 weeks a year in your particular muscle that we're measuring? What if it really is just kind of episodic expression of this gene? People talk about FSH being kind of a stepwise. That I was fine, and then I lost my biceps, and then it was fine, and then I lost my left quad, and then it was fine. So if you're in a clinical trial and you're doing this, then if you're in a plateau phase, then I think that is a real challenge for clinical trial design.
I think that's one thing I heard from the NIH Rare Disease Day, that people that I was talking to said the hardest part about doing a trial on FSH is you can take 100 of us, whether we're related family-wise or not, and none of us will have the same progression, or even in our own body. You know, your right leg is stronger than your left leg, but your left leg is stronger in another way. So that's everybody I spoke to said that is the biggest challenge of trying to do a trial with.
Yeah, it really may be. You know, if we have a drug that makes people stronger, that doesn't just halt the progression of the disease, but that makes the muscles stronger, then that's something that we can study no matter what the progression is. But if we have really episodic progression that varies hugely even within an individual, and we're just trying to stabilize, Robbie Tweel has done an analysis that shows you're going to need hundreds of people to get significance. Any questions online?
I haven't received any.
They went back to sleep in their pajamas.
They're actually the max of the day at the moment. There are more people watching right now.
Okay.
Could you repeat the creatine and CoQ10 amounts that you had?
Yeah, so you're going to have normal kidney function because creatine is excreted through the kidneys. And if you're an adult male, your dose would be 10 grams a day. And coenzyme Q10, there's not a clear, but I would say somewhere 300, 400 milligrams a day. So 200 twice a day would be a great way to take coenzyme Q10 for an adult male.
Thank you.
Any interest in testing for minerals like magnesium? Would that be an issue with muscles? You know, deficiencies, I'm saying.
Yeah.
And you talked about the D3, you know, seeing that across the board is
low.
Is low.
Right. So the question was, should we be screening for various deficiencies? And I don't know what we should screen for that would have a direct relevance to muscle. Other than what we usually have in our metabolic panel, which does frequently include potassium, magnesium, sodium, fluoride. Those are not necessarily, what did you call them, metals?
Minerals.
Minerals, right. But on the other hand, if people didn't really realize vitamin D3 doesn't not only helps your bone, but helps your immune system, helps your muscle until fairly recently. So, you know, we'll continue to learn about that. You know, I think it's extraordinarily interesting the wide effects of vitamin D3 that are just really starting to become known.
I'm not sure you'll be able to answer this, but I'm just curious to know, like, when you think about a clinical trial or an observational trial, what kind of budget do you need to have in place to fund it? And I'm sure it varies widely, but
yeah,
that might help the organization think about targeting specific fundraising projects.
So the question was, what's the budget to run a clinical trial? And for a drug trial, let's say a multi-center drug trial like MY29, the budget is around $10 million. For a single-site trial, you know, much more limited. For instance, we did sildenafil in Duchenne. You know, the budget is more like $500,000. But if you're going to be enrolling around 100 subjects at multiple different sites, it's in the millions. All right. Well, Jenny, did you want to say a couple words about what our studies, or shall I just mention them again? Since
Well, for FSH,
yes.
We have the Hi, I'm Jenny Vivat, and I'm Dr. Wagner's clinical research coordinator.
That way.
Okay.
There you go.
Oh, that way. So anyway, we have several studies for FSH.
The first one and the longest running is the NIH-funded FSH biomaterial for Wellstone 4. We are collaborating with the University of Massachusetts, and basically it entails FSHD patients genetically confirmed and their first-degree unaffected relative. Right now, our focus before it has just been, blood collection and muscle biopsy. But currently, we have amended the protocol to include those that are younger, like age 2 to adulthood, and they can either have a muscle biopsy or a skin biopsy, and then we also collect blood samples. So we would like, we share those samples to our collaborators in UMass, and they culture and keep the samples to be shared to the other FSH investigators. Our second study is Dr. Leon's MRI study, which currently includes whole body MRI, and she's scanning controls and FSHD positive patients. There's also a follow-up study for this particular research protocol. It's currently recruiting patients.
But we're really trying to focus on kind of a very kind of specific group of people. One is that people who have already participated in the study and bringing them back to get follow-up imaging, and just to see how things have changed over time. And also, we talked about these groups of asymptomatic or non-symptomatic individuals who have a gene mutation that is supposed to be predictive of FSH. And I am really, really wanting to scan more of those. And so those are just sort of based on kind of where we are in the study. Those are the people that I would really like to focus my recruitment on.
Our third study is the one that we're currently boosting our recruitment and enrollment, and that's the bone health study for FSHD patients funded by FSHD Global. So this study involves neurological evaluation, blood collection, and DEXA scan. So it's a one-visit study participation, and we hope to enroll 50 patients. And the group in Australia is also doing the same thing with 50 patients. So the aim is to determine the bone health status of FSHD patients, and the endpoint is, of course, to establish guidelines in the diagnosis and treatment of osteoporosis, osteopenia in FSHD. And then we also have one other study, and this is for a collection of data where we do functional assessments for those ambulatory and non-ambulatory FSHD patients. And the aim, of course, is to determine a good outcome measure for future clinical trials. And hopefully we'll have a clinical trial for FSHD soon.